Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Claudio Cantù from Linköping University on the show. Please let me briefly introduce you to our audience. You obtained your PhD from the University of Milano, and in 2011 you joined the Developmental Genetics Lab of Professor Dr. Konrad Basler at the University of Zurich, Switzerland, for a postdoc. Then in February 2018 you moved from Switzerland to Sweden, where you started your own research group, and you are still there today as a senior associate professor. A question I like to ask every guest to start off our podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
All right. I'm glad I received this question as a first one. I would wonder how someone could not be interested in biology in a way. And I think that I was interested in biology as a child, primarily, and this thing never went away. And then as a child, I was typically interested in dinosaurs and sharks. But then something happened when I was 19, 20, and I studied genetics at the university. When we were studying the experiments in, the genetics experiment in Drosophila melanogaster at the beginning of the 20th century, from Thomas Hunt Morgan and his young students. And somehow, in that moment, I think I switched my interest to the kind of green biology of animals, to molecular biology and genetics. And I think that what most fascinated me was that we could understand in details things that are extremely complicated, but which we cannot see. We cannot even see in principle, like DNA as a size that is smaller than the wavelength of light, but yet we understand how its structure and how it works. And this was just, I think, amazing. So in that moment, I think that I wanted to study marine biology or ecology, but then I switched immediately to interest in genetics and molecular biology, and this never went away.
Coming to a science that centers around the mechanisms via which cells influence each other's genomes during development, which is very broad. This episode might be a little bit different than others because I hope we can have an open discussion, and I want to get your opinion on a topic that might be interesting to many. Currently, there are 2 papers on bioRxiv, and this is also how I became aware of you and your work and wanted to invite you to this podcast with the title The CUT&RUN Blacklist of Problematic Regions of the Genome and Exhaustive Identification of Genome-Wide Binding Events of Transcriptional Regulators with ICEBERG. So let's dive into it. But let's start with your work optimizing CUT&RUN and calling this CUT&RUN Love You. Can you talk about your motivation or the need to modify the original CUT&RUN approach and what advantage this method brought then for you?
Yeah. This is very interesting for me, at least. So when I was a postdoc at the University of Zurich in the lab of Conny Basler, something that I was working on mechanisms downstream of Wnt signaling. So I wanted to look at what are the genes that beta-catenin, the sort of the fulcrum of Wnt signaling, regulates directly. And then I was doing ChIP-seq on beta-catenin. So ChIP-seq, like the gold standard technology to look at transcription factor binding genome-wide. The problem is that beta-catenin maybe has a very dynamic way of action, or it doesn't directly bind the DNA, or for some reason it was very difficult to detect with ChIP-seq. And then I spent basically 2 years of my life as a postdoc to kind of use information from other people who were successful with this and try to adapt a ChIP-seq protocol and profile beta-catenin binding from in vivo tissues in the mouse. And then we were successful to some extent, in the sense that we got data sets which were good, quite noisy, but sort of also the reviewers accepted them because everyone knew that it was a very difficult target to.
So what were the factors that you optimized? Is it the fixative, the fixation time, or what did you change?
Yeah, so there was a lot of choice of antibodies, and I think that the tricks that made it work had to do with the amount of cells and with the cross-linking quality and quantity. And for this, I was inspired by a paper from the Hans Clevers group. And then what really worked was the combination of two chemical cross-linkers. One gets usually use this formaldehyde, which cross-links everything with everything, and adding another one, which is specific protein-to-protein cross-linker. And this very likely made the experiment working, because then beta-catenin doesn't bind directly DNA, it relies on other transcription factors to do so. So then we were cross-linking beta-catenin to those proteins that then in turn bind DNA, and which are cross-linked likely via formaldehyde. The other one is the trick that everyone knows in the ChIP-seq world, which is using as many cells as you could. Which, however, was also a problem because I aimed at working in vivo using mouse embryos. And then what me and Dario Zimmerli, PhD student at the time at the University of Zurich, were doing, it was an hecatomb. We had to sacrifice a lot of pregnant female and sacrifice many hundreds of embryos to do a single experiment. And this also, of course, poses like the ethical dilemma. It's like, is it worth it? So from there, then I moved in 2018 and I set up my lab at the University of Linköping in Sweden. And I wanted to have some sort of a ChIP-seq centered lab. The reason for this is because I think it's a primarily kind of an interest and passion-driven choice, in the sense that I really like to see the gene regulation events at the interface between the regulators, which are the transcription factors, the chromatin regulators, and the DNA, the genome. And I also think that experiments like ChIP-seq or CUT&RUN now allows you to have the highest resolution of what we define being a molecular mechanism, which is always this vague concept that journals and editors and reviewers want from you. So basically, at that time, I wanted to establish ChIP-seq based projects. And then I have read the Henikoff paper in eLife about establishing the CUT&RUN. And I thought about this sort of dual advantage that CUT&RUN has, which is it doesn't require cross-linking, and you can have a ChIP-seq-like profile, but with very small number of cells. And I thought, I need to change immediately. It was not an easy choice for me, because at that time I had already invested quite some money. For example, I paid $50,000 of my initial grant to buy a Covaris ultrasonicator, which is a great instrument required for ChIP. And the choice would make this instrument almost obsolete. But then I thought it might have been a winning choice, and I think it has been a winning choice. The problem was that I suffered to tune ChIP-seq for beta-catenin, and then when I was working with my initial co-workers, like, for example, Matthias Pernebrink, who is now at the university, is at the Institute of Molecular Pathology in Vienna, we couldn't make CUT&RUN work on beta-catenin. So I thought that my former investment and my current investment of moving from ChIP-seq to CUT&RUN was the wrong one.
When you do like massive cross-linking and this made it work, right? And then you move to a method that omits the cross-linking. I mean, it could be difficult, right?
Yeah, it could be difficult. Yeah, maybe with hindsight, it was probably obvious. And I don't know what are the reasons why we insisted rather than going simply back to ChIP. maybe a little bit of serendipity, maybe also the persistence of the person who was working with me at the time, Mattias, and then Gianluca Zambanini, a fantastic student from Italy. So they were persistent, and they found ways to make CUT&RUN work for beta-catenin. And then we kind of established this slightly modified protocol. And what I liked about their work was that they really thought through the reasons why the experiment targeting beta-catenin was not working. And they tested those hypotheses until they found the one which presumably was the culprit cause of why targeting beta-catenin with CUT&RUN did not really work.
What was the key effect that made it work ultimately? Because I think what this is like the problem with some of the transcription factors that are not working in CUT&RUN and CUT&TAG is might be the reason why beta-catenin then worked in CUT&RUN.
Yeah, exactly. So let's say that we had a few hypotheses of why beta-catenin could not work. My original idea was that beta-catenin, being relatively distant from DNA as it doesn't bind it directly, would not allow micrococcal nuclease that use in CUT&RUN to cut.
Oh, because it's too far.
DNA.
Because it's too far away.
It could be. I thought it could be too far away. The PA-MNase doesn't reach DNA when it's directed to beta-catenin. For example, we try to solve this by using primary and secondary antibodies. We thought we extended the reach of PA-MNase, and this never really gave us good results. What ultimately could give results, of course, was using nuclear extraction, because beta-catenin goes into nucleus, but there is quite some beta-catenin, of course, on the membrane and then the cytoplasm, and we thought this titrates away, binds the D, and sequester the antibody. But I think that the main leap forward to positive results was when we started doing in situ denaturation of proteins. So at the end of the CUT&RUN procedure, when you are meant to elute your DNA fragments before you do library preparation for next generation sequencing, we worked under the assumption that maybe beta-catenin is in the relatively big transcriptional complex, and the DNA might be cut left and right to the transcription factor or the transcriptional complex, but it's somehow trapped into the protein complex. So we started giving urea to this step in the reaction, and this, as a matter of fact, resulted in a huge yield of bigger DNA fragment. Bigger DNA fragments that were released, and when we're collecting them, they would map to the binding profile of beta-catenin, which we knew already, of course, from, for example, ChIP-seq experiments that we have done. So this allowed us to identify that basically if you do in situ protein denaturation, it really looked like that those big protein complexes trap some DNA. So some DNA remain like stuck within those. And then you can release them by simply denaturating the complex. So this is now our favorite explanation of why the experiment then worked.
The primary antibody in the MNase would not cut or be or would not go there where the protein is binding, but that the complex after digestion would not release the DNA that you ultimately cut.
Yeah. This is what we now think was the case, because it was urea who gave really this leap of release in DNA fragments.
Interesting.
And of course, we could also measure that the size of the fragment released with urea is typically larger, and this sort of gives an intuitive sense that this might be the case, that those are more likely remaining trapped into protein complexes and so on.
So the ENCODE project and also others have compiled a blacklist for ChIP-seq, which have been widely adopted. These lists contain regions of high and unstructured signal, regardless of cell types or protein target. And you set out to do the same for CUT&RUN. So what was the initial situation you found yourself in when starting with this?
Of course, we have been using every time we do a CUT&RUN experiment, then we have been using the previous blacklist.
So for CHIP?
That is used for CHIP, because the reasoning is that those regions, the regions that are present in a blacklist have an obscure origin, in the sense that many are recognizable as regions of the genome that are difficult to be mapped in principle because they are highly repetitive, and they can be subject, for example, to also mapping artifacts. So you can have a few duplicated regions or repetitive regions, and then many of the amplified fragments in your sequencing library map there, maybe as an artifact, and so on. And then what you see, as a matter of fact, is a signal that looks like a peak. But then you would see this signal also when you do a non-antibody experiment, or an IgG control, or a non-related antibody targeting something that is not related to your biological hypothesis. You would see signal there. So then, very wisely, I believe that someone had the idea to compile a list of regions that it's good to discard because they show signal. As again, the ChIP-seq blacklist might have included many types of artifacts that are mapping artifacts, that I suggested before, but they could also have been, for example, cross-linking artifacts. We cannot exclude that when you apply cross-linking, there are maybe big protein complexes somewhere in the genome that have nothing to do with the protein that you're trying to pull down, yet they get cross-linked, and for some chemical reason they are preferentially, or they are enriched also in your pull down. So you find signal there. So many are the reasons why you get spurious signal, and they were very wise in compiling this ChIP-seq blacklist. And we have used those dataset to subtract it from our datasets. But then we realized that, so then we have many projects in the lab that essentially are CUT&RUN centered. And we realized that many of those regions that are spurious signal and identified in ChIP-seq blacklist are also appearing in the CUT&RUN experiment. So we thought it's wise to remove them. But then in particular, Anna Nordin, now she's a PhD student in my lab, she realized that there were other regions that appeared often in our CUT&RUN experiments, but they were not included in a CUT&RUN, in a ChIP-seq ENCODE blacklist. So I have to admit, entirely on her initiative, she compiled a list and she proposed this project to me. Of course, I was very enthusiastic because of the importance of the project. I mean, we all need to rely on the good tool made by ENCODE, which, however, for CUT&RUN, it's not complete, because CUT&RUN identifies new regions that are also found when we use an IgG sort of pre-immune serum, like a mixture of unrelated antibody as a control. So we thought it's important to tell the community that those regions should raise suspicion, at least.
So to your knowledge, are you the first and only one working on such a blacklist?
No, I know that there are persons who might have been looking at compiling other blacklists, and in fact, the bioRxiv paper was now cited by another group who I think has similar goals. But I believe that we were the only one trying to build the CUT&RUN specific blacklist. Yeah, to my knowledge, this is the case. Of course, we are trying to be in contact with the CUT&RUN community, and I hope that they find our work useful, and to build the CUT&RUN blacklist, which we now are considering changing the name, also upon suggestion of the editor, in which the paper now, the bioRxiv article is now in a revision, is to change the name from blacklist to suspect list, also based on the rationale, which I somehow like, actually not somehow, very much like, the rationale that our regions that display signal also when you use IgG antibodies, it's difficult to conclude that they are wrong signal. For example, some of those end up in promoters. And we cannot, and even if we use no antibody or IgG, so there is a problem in those regions. But we should warn other users that they might be the scientists working on trying to target the factor which maybe regulates those promoters. So people should take also all the blacklists with caution, because we cannot exclude that any region of the genome, it is actually regulated by some rare factor. Mm-hmm. Yeah, this is the story of our work, and we still use it now. After having compiled this, we have done literally hundreds of CUT&RUN, and we always use this tool, which is made up by. So we have done like a reiterative experiment with IgG control, but we have compiled, of course, the blacklist using dataset from others. We don't want to bias our blacklist with our own hands and experience. So
this was to see. Because many questions I wrote down, you already kind of touched upon, but so how was the technical approach you took? So you took like data sets from your own lab and then also from other labs, and you kind of bioinformatically compiled them together and make kind of an average of all the peaks that might come up in all of them?
Yeah, precise. This was precisely what we have done in a nutshell. So originally, like historically in the lab, we had done several IgG controls, and we started compiling blacklist using those. And we noticed that if we would take 20 IgG controls that we have done and looked at all the peaks that are called, let's say, for example, in a majority of the experiments, then we would have a list of suspect regions that appeared in our own experiments. And if we used, and then let's say call our lab blacklist, and when we applied this to other datasets, we could see that those datasets were improved, in the sense that most of the dataset also included those signals, which then, of course, we always rely on the fact of specificity. So we trust the peculiarity of our discovery when that signal is not found in the control. So in the very moment in which we notice that across the board of our experiments targeting our favorite transcription factors, the signal that we had identified in the blacklist that we had compiled was present, then we felt the urge of excluding it. This might be genuine good signal, but because it appears also in the control, it's safer to exclude it. So we want to remove false positives. And on the false positive concept, there might be a twist if we are curious to talk about the ICEBERG.
That's later down the road. I also wanted to get in, how does the CUT&RUN blacklist compare to the ENCODE blacklist? So is there something that come up that is maybe interesting? Or is it just like a list of peaks that does not tell anything more interesting than just using it?
Yeah, this is like, I would have to go in the wild world of speculations here, because there are peaks overlapping with the ENCODE blacklist, but not all of them. So the CUT&RUN blacklist doesn't identify some of the ENCODE blacklist, and the CUT&RUN blacklist is smaller. So it's a set of a smaller set of peaks, and many are in regions that are obviously, when you look at the sequence and you're in a centromere and then sequence, there are a lot of repetitive regions, and there are a lot of peaks there that appear across our IgG experiments and across the control experiments of the other published data sets from independent laboratories. And those sequences don't tell me much, but of course me is a person with limited ability to see through DNA sequencing and regulatory regions. Some of those regions, as I said, I believe we have, for example, one blacklist or suspect list peak in the GSK3 promoter, which is of course a very important gene. And then we get this peak across our experiments, and we have to exclude it because it's present in the blacklist. However, we wish to warn caution in the sense that someone is bound to identify a factor which regulates GSK3, which is a gene that must be regulated on the promoter by transcriptional regulators. So also this is why I favor also the change of name from blacklist to suspect list, because I think that scientists also have to use those lists with cum grano salis, like a little bit of caution, and look at those instances individually in each of your experiment.
Yeah, I mean, it would be interesting to see if there is a regulation at the promoter, does this blacklist peak change indeed, right? I mean, if it's still the same after there is indeed a regulation, but it still shows up, it might be dangerous for the interpretation of this specific experiment then.
Yeah, of course. And the other aspect maybe that to follow up your question about what we learn about on those blacklists is that this also upon I think a very genuinely good suggestion from a reviewer. We have looked at, and then so this will be published in another version of the paper. We have looked at the motifs that are the transcription factor binding motifs of the regions that we identified in the blacklist. And there is quite a lot of things going on. So you can think, for example, we identify CTCF motifs, and then you can think, well, is it CTCF binding that produces spurious signal and so on? It doesn't look like the case when we, for example, overlap the blacklist with our CTCF binding profile. And also when we looked at, so we have done the same analysis of motif search in the ENCODE blacklist. And also there we identify quite a lot of transcription factor binding signatures. But I don't think that we can learn much from this for the reason that motifs are degenerate DNA sequences. So a motif could be a bad motif and does not really allow stringent binding of transcription factors. And also motifs are short DNA sequencing. So the more you expand the pool of the DNA sequencing you look for, you are screening, the higher the chance of identifying by mere luck some motifs. So I don't think that you necessarily learn something from those. The ultimate test would be is that region actually bound by that transcription factor with that motif, and then we need to do a ChIP-seq or a CUT&RUN.
Yeah. So, sorry to conclude this section. Can you say something? And if you can't, that's perfectly fine. When this will be peer-reviewed and published? Because now it's on bioRxiv, but when this will be listened to in like a year from now, then it may be out somewhere.
Yeah, it will be out. So it was. I believe I can say this now. So accepted as an article, and it will be published, I think, in a time frame of a month or something like this. And we've included all the peer review, and mostly it was, I would have to say, great suggestions. And then we were happy to have also. This is a case in which, when I grew up also as a postdoc, I started being a little bit against the peer review system. They are just simply denying me to publish quickly my papers, which are correct. And then, of course, in many instances, I had to revise my opinion on it. And this is one of those cases in the sense that I grew up as a molecular biologist and a developmental biologist. And those projects, which are almost entirely computational, like Anna, who is the leading author on this and working in my lab, and myself really wanted expert opinion on this. I mean, just please help us to understand if you're doing something wrong. And the very fact that, let's say, at least 2 reviewers out of 3 took the project with enthusiasm, then this made us also happy and like more anxious in going ahead with the application.
So next to this blacklist project, another question that might come up or has come up in the one or the other experiment is how many peaks are overlapping with GPseq experiments, and how is the overlap of peaks in biological replicates in CUT&RUN? To solve this, you developed ICEBERG, and this stands for Increased Capture of Enrichment by Exhaustive Replicate Aggregation. An experimental and computational, again, a procedure that utilizes numerous CUT&RUN replicates to discover the entire set of binding events and consistently exclude false positives. So maybe to start off, a really easy question: when is a peak really a peak?
Yeah, this is a $1 million question whose answer, in my opinion, is no one knows. No one really knows. And what we have to do, and the reason why I say this is because the peak is identification of signal in a specific genomic coordinates. Now, as it turns out, for example, when you do CHIP, in CHIP you are purifying cross-linked genomes. And then you hope that via immunoprecipitation, you are enriching for those DNA regions that are bound by your transcription factor. Enrichment precisely means that there will be a ratio between the signal in that position and the left and right adjacent sites.
The most famous signal-to-noise ratio, right?
Exactly. This is the signal-to-noise ratio, which this needs to be higher than 1, in the sense that if the signal-to-noise ratio is 1, in that position, your signal is equivalent to background, so you cannot draw any conclusion. So is the signal-to-noise ratio 1.1, 1.2, 1.8, 2.5? Where is the number where we trust it as a signal, as long as it is reproducible? This is a genuinely difficult question that pertains to the signal detection theory, of which I cannot say more in terms of math, but I only have an intuitive understanding of it. It's a problem of detecting anything, even if you are piloting a plane and you have a radar and you spot a signal. This might be another a fighter jet trying to bombard us, or maybe it's a flock of birds, and then you need to distinguish these 2 signals. And this is a genuinely a problem in all fields of human enterprise, where we need to detect something that is not something else.
I mean, it's a problem of measurement, right?
It's a problem of measurement, exactly. So how people deal with this in ChIP-seq, I think it's still the best way, is 2 ways. One, statistics. We use statistics to impose thresholds, and then you can imagine this is a line just above the signal, across the entire genome in a ChIP-seq or CUT&RUN experiment. Signal that goes above, we call this is a peak. Signal that goes below, we call this is not a peak. That's a great method. The other method is replication. We do the experiment again and again, and then we say the peak in that position happened again, so I trust it. And of course, those are the 2 methods on which everyone is relying, and I have been relying, and people should be relying also now, I think. On the other hand, there are problems, like if you become somehow in inverted comma an expert in ChIP-seq or CUT&RUN, in the sense that you are used to walk through the genome and scroll the genome to see this is a peak, this is not a peak. You sometimes recognize this is genuinely a peak because of its shape. It really looks like a ChIP-seq peak, where there is a peak summit, and this is the obvious result of the biological procedure when a transcription factor in the position that is close to the peak summit is binding. Then this looks like a genuinely good peak. Other peaks are obviously not good because, for example, they are big squares, like tall squares, and they are obviously the result of PCR amplification of this library species of DNA. But then often what you see is that, come on, that's really a great peak. It's called the cross replicates, but it's always a little bit below the threshold, so you don't call it. This is a common problem that everyone, I think, in the field knows, and you have to exclude it. But we are okay in excluding it because what we write in the paper. So scientists don't like being wrong. So if you have a positive signal, but we are not entirely sure, you just exclude it. You are okay to have false negatives. So that's a negative, but it could actually be a positive, but it's okay to call it as a negative. What you don't want is false positive, because false positive means I go around on the street, publish my paper, but then I turn out to be wrong. So we don't like that. And there is this sort of unwritten rule or common wisdom across scientists that we are okay with the false negatives, but not okay with false positives. But then we started thinking that, like, if I am a patient and I go and I have a pain here in my stomach and I go to a doctor, I would rather pay the cost of a false positive, in the sense that if I have a disease, but the doctor doesn't diagnose it, this is a false negative, then I end up having a bad disease that is not diagnosed and I don't know it until it's too late. While I'm okay to risk a false positive, so like increase the detection rate of your tool, because then I will pay the cost of a second opinion or a second analysis to make sure. So the replication, I would want to rely on the replication rather than the fact that my disease is not detected. So we were wondering whether this strict preference for false negatives over false positive is an idiosyncrasy of molecular biologists. I mean, we work in this field where this is the case, but is this a right attitude in all fields of human knowledge? Of course, we don't know the answer to this, but we sort of provocatively say, shall we try to be daring a little bit and maybe welcome some more signal and find another way to detect whether the signal is genuinely good or not? In other words, we are okay to exclude peaks on which we are not sure, but we know that we identify what...
You worked hard enough to say that it's not a peak because you did some other controls.
You did other controls, but sometimes you are left honestly with a doubt, and you have big data sets that you need to exclude because they are within the set of unreplicability. If you imagine a simple Venn diagram with the two sets, the intersection set is what you trust. The two non-intersecting sets is what you say, I don't trust those. But those sets are often big, and this made us think that the intersection set, which is our discovery that we trust, might represent the tip of the iceberg of the biologically relevant events. And then we thought, well, I want to discover the beta-catenin binding profile, not the tip of the iceberg of beta-catenin binding profile. I want to discover all the binding profiles of beta-catenin. How do we do that? And then we thought that we should try to see what we can say from these two big non-overlapping sets. And then we improved replicability and computation by designing this iceberg, which was essentially designed by, again, Anna Nordin, the person who compiled the blacklist, and also Pier Francesco Pacella is a senior scientist in the lab, and Gianluca Zampanini, who is the original author of the CUT&RUN LoView protocol. By the way, LoView was a real acronym. It stands for low volume and urea. It's not only fun things.
So the term you coined here was peak concordance. And if you compare the so what does it exactly mean? And how does this compare then between, like, again, CUT&RUN and ChIP-seq?
Yeah, I'm actually happy you asked that because while writing the article, which we wrote as a team effort, the article entirely, we were writing, of course, overlap or reproducible peaks. But from a almost philosophical point of view, defining what is reproducible is precisely the problem we were trying to address. So we thought of using a new term to address the overlap, and we agreed on concordance, which is, it's somehow a neutral term that can define the outcome of our technology that spots events more than once across different replicates. And the different replicates are concordant to the extent to which they reproduce those events. So we thought that concordant can become a clearer way to define this identification of peaks that are reproduced in those experimental instances, which doesn't necessarily imply reproducibility, because reproducibility for us often is a synonym of reality, a synonym of that is what our real discovery is. While the concordance could also be due to technical issues. If we don't subtract the blacklist or suspect list, those peaks will be concordant, but not because they represent biological events. They are the result of technical artifacts, most likely.
Yeah. So how is then how did you approach that? I mean, there is this one issue that you have a high peak concordance in CUT&RUN and a high peak concordance between CUT&RUN and, for example, ChIP-seq, right? So how is this reflected, or how did you see that?
Yeah, so it's difficult for me to compare ChIP-seq and CUT&RUN in the sense that we didn't do this analysis thoroughly. What I can say is that historically I was doing ChIP-seq, and the maximum number of replicates I have done for a study for a transcription factor is 3. And I had always noticed that the overlap across replicates is relatively low. Like, and with the low, I mean with the factors such as beta-catenin, as my favorite protein for a number of years, it can be 10 or 20%. Now, why you do one experiment and you get 5,000 peaks, and then another day you do again the same experiment and you get 5,000 peaks, but of these 5,000 on the left and 5,000 on the right, only 1,000 are in common. The reason why this happened has never been clear to me. But then, of course, I was bound to just trust the overlap. And what I would typically choose to do is describe the figure and say, this is the result, 5,000, 5,000, but I trust the high confidence 1,000 peak that overlap. When we were doing CUT&RUN, the problem was similar and quantitatively on a similar scale. Now, so we had 5,000 peaks of CUT&RUN, 5,000 peaks of CUT&RUN in the second replicate, only 1,000 more or less would overlap. I'm talking again about beta-catenin or some difficult targets, because when you target, for example, is something that we show in the ICEBERG study, when you target chromatin marks like post-translational modification of histones or other very stable transcription factors, the overlap is much higher. It's never 100%, but it's much higher. But then the overlap also with CUT&RUN when targeting difficult targets like beta-catenin is typically low. What I don't know, and what I meant at the beginning of my answer, was the non-overlapping targets in ChIP-seq. I don't know how overlapping are those to the non-overlapping targets of CUT&RUN. This is what we didn't find. We didn't look at thoroughly. But the reason why we didn't look at thoroughly this is because there is no ICEBERG equivalent, to my knowledge, of ChIP-seq. which is what we would need to compare the two technologies.
So how many replicates would you then need to confidently say, well, this is a peak for, let's not say maybe histone modifications, because you just said that those might be better or more stable or more often called in different replicates. But if you look at a transcription factor, or let's say beta-catenin, how many replicates, be it technical or biological, how much is enough?
So we don't have a definitive answer on this, but what we could clearly show is that by doing a reiterative replication of your experiment, and this was shown mathematically by Pierfrancesco Pagella in the lab, that basically the discovery curve at some point plateaus. When you go in the case of beta-catenin, when you go over, and we have evidence for at least another factor that this is the case. When you approach the 20 replicates and you go over 20 replicates, for each additional replicate, the discovery rate becomes quite low. So Pierfrancesco was calculating the derivative of the curve, which approaches 0 at the end. And this shows that you have plateaued your discovery, which in our metaphor, we have seen the entire iceberg. Only having this tool allowed us to benchmark sort of the full discovery rate, the iceberg, the full discovery set, with what you would discover if you were to do only 1, 2, or 3 replicates. And in the article, we provide an entire figure for this analysis, because we think that, first of all, CUT&RUN is a relatively expensive and demanding experiment. I think that it's reasonable to expect that people would want to replicate their experiment 2 or 3 times. And then what do you get with that? So you get part of the iceberg, not the entire iceberg. I think that's entirely okay. You discover the most important regulatory events, or maybe the most common ones across the cell population that you are investigating, and so on. And for example, what we have seen is that when you target beta-catenin, if you do 1 replicate or 2 replicates, your discovery rate could be relatively good, depending also on the peak calling strategy that you design. So we looked this in terms of recall and precision, where recall is the number of peaks that belong to the iceberg peaks that you actually identify. So the number of real positive across the total number of positives. And the precision is the number of how precise is your data set is, which means across the all number of peaks that you identified, how many are real positive. And then this really allows you to chart the line between false positive and false negatives. So we have seen that, of course, there is always a trade-off between precision and recall. And regardless of the statistical test that you use, you favor one or the other. But, if you look at the figure in the paper, you can see that even if you do one experiment or 2 experiments...
It's figure 2, right?
It is figure 4, the one to which I refer. Okay. And in those charts, we see that the chart, this is a very simple chart. Admittedly, we were jokingly inspired by the presentation of the first iPhone by Steve Jobs, when it made the chart about the smartphone existing, and iPhone is right on top on the right top part of the chart. And this is true for the iceberg. So the iceberg is the top right part of the chart with perfect precision and perfect recall.
So sorry to interrupt you, but could you just give a short summary about iceberg, because we didn't touch on how it works?
Yeah, you're right. So basically what iceberg is a combination of experimentation and computation, where we did reiterative replicates, and we did 25 replicates of CUT&RUN for beta-catenin. And then we, for each replicate, we selected a random, at random reads for each replicate, and put them all in one bag, such that to build a big individual replicate, essentially, that includes information from all the experiments. And the reason for the rationale behind this was that the spurious signal, which could be due to random effects of mapping or purification of specific DNA fragments, would be averaged out if you sum that coming from a large number of experiments, such that your background becomes a more flat line across the genome, while the small signal, even if it's small and typically falls below the threshold of detectability in individual replicates, if it's real, it might be summed and accumulated such that it would be above the threshold of typical peak calling statistics, and we might identify it. And this actually is what does happen. And we also try to explain the iceberg logic in a drawing. And I think that this is really the case, that the background became a tamer background, in the sense that we can really understand it better because it's more flat, while the real signal appeared because this was the result of summing up all the small signals present in some, but not in all individual replicates. And then basically this allowed us to discover several peaks that were called maybe in a very small number of replicates. For example, we do 25 replicates. If a peak is present in 25 out of 25, that's an easy choice. We like it. If it's present in 20 out of 25, kind of we like it. It's like 4/5 of our experiments display that signal. This is quite a high chance. But then if you start looking at numbers of the signal present in 10 out of 25 experiments, do you trust this? This is a minority of experiments, but still, CUT&RUN is a highly controlled experiment. You can detect that each experiment worked. And then this signal is present in 10 times. 10 times is a high number. Typically, no one does CUT&RUN 10 times. And then, so we thought that we should trust those. And we should also trust signal that maybe appeared in even a smaller number of replicates. And we started thinking that maybe the frequency of signal detection across replicates might simply underlie the probability of that factor to be identified there. And I think that this is an important concept because it's something that people say in the ChIP-seq field for 20 years now, which is the peak is bigger when the binding event occurs in a big portion of the population that you are on which you are doing the experiment. Yeah, if for example, it's possible that cells are heterogeneous. If a transcription factor is binding on promoter of gene X only on 10% of the cells that you are purifying, then the peak will be smaller. These people have this feel since 20 years. And I think that with iceberg, we capture this probability that could be reflective either of the fraction of cells in which the regulation event occurs, or it could be reflective of the time that the transcription factor or regulator spends on the binding there. And of course, we don't know what of these two possibilities, but I think it's in my opinion, is a beautiful hypothesis that demands, like, screams for future investigation.
I mean, having looking at the first point you just raised, so it's only single cells that might have this factor bound at a given time in this experiment, and then a single cell experiment would obviously be the right experiment to do it then, right?
Yes, of course. it is, of course, the single cell approaches are, I believe, the future, and I hope that we don't have to wait too long. Now there are approaches to do, in particular, to apply cut and tag to single cells, and they've shown to be reliable and yeah, I mean, you're entirely right. That this would be the ultimate approach. Of course, when you look at single cell approaches, if you look at attack sequencing or cut and tag on single cells, and if you have one cell, then you have per each cell in that specific chromosomal position, this becomes a digital problem in the sense that either you have signal or you not, or you have your two alleles, so you might have zero signal, signal 1 or signal 2, because you detect the binding on 1 or 2 alleles or 0. And I think a challenge in the future will be also to address what does reproducibility mean there, because when you are relying on digital events to call out cell identity, so this cell, the blue cell is different than the red cell because it presents this digital event, then you have a problem of reproducibility, because we want to make sure that something is true if that event occurs again, while here conceptually we are distinguishing two cells because of an event that is individual for that cell. So when the events are digital, I think it's going to be, of course, not impossible, but more difficult to distinguish a real biological idiosyncrasy defining cell identity.
Yeah, I mean, then other factors might come into play, like cell cycle and other things, right? Why is this vector here? Yeah. Why isn't it here? Right? So,
yeah. But of course, the singles, the point that you raised, there is no better answer than saying you're right. And I also look forward that the people who are working hard on this will develop the technology that allow us to detect, to do a single cell iceberg, for example.
So, just to finish off this part, what would you say? How many biological replicates are enough? How much or how many should a researcher be doing?
Yeah. So,
I mean, nobody can do 25 as you did, right?
So, well, people could do 25 in the sense that if, of course, not everyone maybe could afford doing experiments, but if you consider the sequencing costs and now the also reduced cost of library preparation for next generation sequencing, now we are in the lab, we are often confronted with the choice. Should we do CUT&RUN qPCR or should we just sequence? And then we decide to sequence because the gain of doing CUT&RUN qPCR is not really substantial. I mean, if we do a full plate of CyberGreen-based qPCR, we pay more than the contribution of those few samples within a sequencing lane. So we would rather wait, accumulate samples enough, and then include them in a sequencing, because their relative contribution is lower than the CyberGreen plate. And of course, but then you have to add the costs of antibodies. I mean, of course, this is expensive stuff, but also if you want to do a staining on like immunohistochemistry or immunofluorescence on a tissue section, then you need to buy an antibody, which costs $500.
But also the cost and the amount of antibody you need for a cut-and-run experiment is way lower than for ChIP-seq, for example, right?
For example, yeah. I'm glad you point this out precisely. So we say in the ICEBERG paper, we say, we don't expect that everyone does 25 replicates. On the other hand, one shouldn't think of this as an impossible feat, because it's 25 replicates of CUT&RUN. I hope I'm not saying something that is awfully wrong, but I don't believe so. It's kind of less expensive than an individual single cell sequencing experiment with the current technology that uses the appropriate controls and the appropriate number of replicates, which are great technologies, and they are expensive. The cost will drop in the future, of course, but nowadays it would cost my lab less to do an iceberg of the next transcription factor than a single cell sequencing experiment, which we do. I don't wanna. So we do, and we love the single cell sequencing technologies. But to go back to your question, I think that's why we have figure 4 in the paper, because there is a world out there of people who are packaging their story with 2 or 3 replicates. And if I'm now to review an article for a scientific journal, I wouldn't say do 25 replicates, but I would urge the scientists at least to try to address the point of what is their fraction of discovery. How can they estimate how much of their own iceberg are they detecting? I think it's very important. I mean, all of us won't discover genuinely good thing. No one of us claims we discovered the entire functioning of the universe. I discovered that this transcription factor regulates gene X and maybe gene Y, and this has these consequences. And then we don't know all the reality, and that's okay. And the iceberg could be really used as a tool to have an estimate of how certain am I about my discovery, and likely how much am I losing it. And that's okay. I think it's entirely fine. And if I may add as a last thing, an important piece of data is that when you target chromatin marks, which is also an important part of the efforts from the community, like there are several chromatin marks and they define how the genome is used. Even when you do 1 replicate, this does pretty well in identifying the iceberg. And we have seen that across 10 replicates, the concordance, the overlap of those replicates, it's very high. And most of the peaks that we identified does occur in all of the 10 replicates. And this is spectacularly showing us that depending on the factor, you really have a different outcome and expectation in terms of how many times you have to do the experiment to be satisfied with your discovery rate.
So just a small question to add. When you say replicate, this is a true biological replicate, right? So you start with a separate biological entity at the beginning, right? So you're not talking about like some splitting up in between and doing some kind of technical replicate, but it's a true biological replicate then.
I don't know. But in the lab, we thought of this very often. This is a discussion that is always open but never closed, because we don't know how to address this point. What is a technical replicate? What is a biological replicate? When, for example, you are assessing you are doing a CUT&RUN experiment in a cell culture. And of course, there are reasonable opinions on that, but how we addressed the iceberg problem was from a technical perspective. We know that because we wanted to be even more precise and not give the chance that other biological factors, which have to do with the temperature outside, that now it's summer, is changing and the cells inside our lab grow more, might influence this. So what we did, we cultured big amount of cells and we split them in. Actually, we did this in 2 rounds, and then we split them in 10 or 15 samples, as such that we can have 10 or 15 of what we refer to technical replicates. And then 2 weeks later, we have done again other 15, and those are 15 technical replicates. One could think that the 2 experiments are 2 big biological variations, biological replicates. On the other hand, so Anna did look at with the classical, for example, principal component analysis, that you cannot really distinguish the 2 groups.
That's a good information.
This was very important. But yet, even when you do technical, the experiment should be exactly the same, but they are not. and we don't know why. And if you look at figure 2, then what we show there with a good dose of pride is all the 25 replicates. They all show that they worked spectacularly well because we have very high signal and very low background in the region where we know there should be signal. And they all look like each other. But if you zoom in, you see that there are differences in peak size also in this region. Why this happens? That's the problem we try to address.
So is this what you are working on like currently, and for maybe the next 5 years, that you're trying to address the technical and biological variability in those experiments?
Yes, in part, not only. So we started off, I as a scientist and my lab as a whole, we started off to address biological questions, not really technical questions. I'm also kind of a little bit, I'm happy with hindsight that we are also dealing with technology and statistical and perhaps a little bit philosophical issues on how to deal with signal detection theory in molecular biology and genomics. So I'm admittedly proud of the people in my lab who really can do that. But we started off with the genuine biological question, which I think it's what we are going to focus on in the future. In particular, for example, we are going to use those tools to address the activity of beta-catenin and other factors which for many years have been shown to operate in somehow a universal manner and conserved across the species, model organisms, and across cell types. But if you look at in details, when we apply like tissue specifically those technologies, we look at proteins that like beta-catenin act in a very different way, and we are trying to understand first how and then why. This is what we are going to focus a lot in the future. But another thing that I learned in my still short career is that what I think is going to happen, it's not going to happen, and some other things will happen, like such as ICEBERG. I never anticipated that we would focus our efforts on such an enterprise.
So, Claudio, this is now officially the longest podcast episode we did, and we have like 106 now. So thank you very much for your time and for being on the show.
I'm very grateful for your invitation, and I apologize. I become very talkative. I realize when it's about
it's great. It was great information that you shared.
But thank you very much. I'm very happy that we did this.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.